Okay, so who has learned about viruses before? You got a little bit? A whole, oh, you had a whole course on virology. Okay. What have you learned about viruses?
And microbiome.
A lot or just a little bit?
Just like a little bit.
Okay. Well, hopefully this supplements what's been learned before. Viruses are very simple and also very diverse and also very complicated all at the same time. So it's kind of a tough topic to tackle in just a couple of lectures. Obviously, people spend their entire lives studying viruses. So there's a lot to talk about. This is the Zika virus. This is a cryo-EM structure. So these pretty pictures are structures of viruses. This lecture is actually really pretty. There's lots of pretty structures. Okay, so just basic terminology. A virion, when people say virion, I wonder if I can draw here. Yeah, there we go. A virion is an infectious particle. So that means when we're talking about biology, people have taken my classes before. I always talk about like there's 4 different types of molecules in living things, which are what? What are the different types of molecules that constitute living things? Nucleic acids. Yeah, carbs. Yep, lipids and proteins. So when you hear the term virion, often that is a mixture of all, if not some. Always, when you hear the term virion, it means there's some kind of nucleic acid in some kind of a capsid, which is what we call a virus or a virion. And many times these virions can be glycosylated, which means they have proteins in their capsid, which can receive like glycosyl modifications, which are sugars. So in theory, they have like some sugars in there. And oftentimes they are surrounded by an envelope, which would be lipids. So viruses can be made of all these things, of course. And when we say virion, that means this is the particle that is sufficient for infection. So it's sufficient for infection and propagation of the virus life cycle. So I make this analogy, I guess it's a bad analogy. Viruses are like donuts with cream filling. And the cream filling is the nucleic acid. So it's essentially like a package stuffed with contents. And the stuff that goes in there, the nucleic acid, is very important for characterizing viruses, the taxonomy of viruses. It's very important for the life cycle of viruses because their nucleic acids can be structured in different ways, and that structure determines the physiology of the virus. So the organization of the genome is extremely important in virology. It's kind of like a central theme. Many different flavors, which we will talk about. Okay, so a virion has its viral genome packaged inside of it. You might hear this term phage. We're not going to talk a lot about phages in this class. Phages is a specific type of virus, it refers to bacteriophages. So bacteriophages are what? So this is the typical like virus you envision. This is a phage. What are phages famous for? Bacteriophage. What would they do? Any idea? Bacter. Where have we heard bacter before? What? Yeah, they kill bacteria. Okay, so phage is a very specific term referring to viruses that kill bacteria. Bacteriophage then is virus that kills bacteria. So this typical phage drawing kind of that you see, the structure, this is usually, I think this is a bacteriophage. And so essentially the important point here is not all viruses can infect all cells. Viruses are very, very, very specialized through evolution, and they can only replicate in very specific host cells. So is it often that humans are getting sick from bacteriophages? No. There was a shake of the head. No. And the reason is because bacteriophages kill bacteria, not humans. Human cells are vastly different from bacteria. Does that make sense? So it's very uncommon that phages or viruses can sort of like cross hosts. They're usually very specialized for a very particular host. And this is important in virology and important when we discuss specifically viruses that are transmitted by vectors. Okay. So the basic Virion structure, obviously this virus looks vastly different from this virus, right? So the structure of viruses can vary significantly, but essentially just keep in mind, it's sort of a package filled with stuff, and the stuff is the genome, okay? So capsid proteins are the proteins that constitute, I'm actually pointing at the bilayer here, the proteins that make up this outer shell. So capsid proteins, if you were to like draw this out, capsid proteins are like these proteins that like often geometrically kind of like fit together to form these shapes. And then these shapes are often filled with the genomic content. So if you hear capsid protein, that's like the outer shell. Some viruses, now, like I said in the very first slide, viruses are vastly diverse. So not all viruses have a membrane. Here you're seeing a virus with a membrane, and it's got some proteins that stick out of that membrane. Not all viruses have membranes, but some do. Some have lipid membranes, envelopes surrounding the capsid. And oftentimes, as I said, there's glycosylation. So that's kind of like represented here by these fancy structures. What would be the purpose of the glycosylation, do you think? In this case of the virus, what would be the point of glycosylating proteins on the outside surface of the virus? What do you think that they would do?
So that when it gets into like another host, the host can isolate itself.
I think you're right, but you're saying it in kind of a reverse way. The glycosylation is very important for the virus to sort of feel where it's going. And like I said, viruses highly specialize in which cells they can invade. And so they're not going to recognize and invade all cells. The glycosylation is often very, very important for them recognizing their specific target, which then they invade and recognize. So it's like a process of touching. They touch the surfaces of other cells, and if they're glycosylation proteins and if they match certain receptors, then they sort of signal this cascade that they can invade. Okay, so our virus is living. Let's talk about, again, this should be sort of like review, especially if you've taken my other classes. There's kind of general things that living organisms do that we define living as living things do these things. What's in this subset of things that living things do? Somebody say something. Yeah, they metabolize stuff. Metabolism means kind of like they're taking in energy. They can build stuff with that energy. They use energy. So in this case, viruses, they for sure like use up energy when they're replicating things, but they don't have a means of like producing their own. So it does not equal, they don't produce any energy. They steal energy. So they're not really living in terms of like metabolism. What other things do we think of living organisms? Growth. Viruses, they don't grow. You think of growth with like multicellular organisms that replicate. There's no real increase of the size. The size is sort of predetermined because of the physical dimensionality of the capsid proteins. So they don't grow. They don't grow. They definitely don't grow. So they're not living according to growth. What else? What? Yeah, usually like movement is considered thing. I mean, viruses would move by like diffusion. So in theory, like diffusion, in theory, like there's some like mechanisms of like them getting around, but they're not really like actively swimming or like seeking out sources of energy or things like that. Definitely not kind of like moving in the classical sense. What else? You guys are missing like the main one. So replication is essentially like all things that are living must replicate. That's sort of the central purpose of all living things. We're sort of like survival machines designed to replicate our genomic content. Viruses do this. Viruses do replicate. They do. But the catch is what? Yeah, they need a host because they don't have all the things that they need for that replication process. So they have to steal the machinery of the host to replicate. Okay, what else? A couple other things. Yeah, I call that adaptation. And I guess there's different types of adaptation in a sense that you could adapt through your genome by mutations, which would be adapting to your environment. They do that. Of course, viruses do evolve, but they don't adapt to like it's like a situational awareness. Like they're not going to change their function based on a certain context that they find within that instance of the virus, within that generation. It's going to take multiple generations for them to adapt. So they kind of do, but not really. And the last thing I have here is like whether or not they're cells. I suppose that's kind of debatable. In a sense, in theory, some of them have kind of like bilayers. And you could consider this like a cell of sorts, right? But they're also not a classical cell in the sense that we normally think of a cellular structure. So they're classified as non-living. They're essentially very, very simple and also yet very complex machines. They're essentially small machines. And it's helpful, I think, to kind of think of them as that. Okay, so the central focus of viruses, interestingly, I mean, they fulfill sort of like the goal of all life. Their central purpose is to just replicate themselves. So we'll talk about their various life strategies and how they replicate. Okay. So let's talk about the process, like the first things that need to happen in a virus as it's going to go through its what we call the virus life cycle, even though we just said viruses are not alive. Let's walk through this. First thing is invasion, okay? And that's going to rely on what I talked about, how they can't just invade like any cell, okay? Cells don't just like, host cells do not just randomly like absorb everything that they contact, right? It's actually like a very sort of specialized process for phagocytosis or endocytosis, the process by which cells are taking in nutrients. And viruses sort of capitalize on that behavior, and they sort of trick the cells into thinking that they want to take them in. So viruses will often have, if this is the virus, viruses will often have coat proteins that will interact with various receptors on their particular host cell. And when they bind, they're going to cause sort of like a tricked cascade. They like hack the system, cause a cascade, which is going to lead to like endocytosis, phagocytosis. And then the virus sort of like comes in. And depending on the virus structure, then the virus is going to sort of like release its genomic contents into the cell and then in theory like replicate. Obviously, this is a little bit of a different process from the bacteriophage, where in this case, there's a classical picture where it's sort of like injecting its genome. So some viruses have invasion mechanisms by which they can actually like inject their genomic contents. Some viruses are more like tricking the cell into like eating them and then just kind of like spilling out into the cell. But that's the first step, is invasion. Okay. What comes next? What does this virus have to do next? What? I think you said it. Go to the genome. Replication. Yeah, so they have to like replicate themselves. Okay, so if we're simplifying the virus life cycle, they invade, they replicate to reproduce their genomes. And then from their genomes, Usually proteins are then synthesized. So then they go through a process of protein synthesis. Okay. And they're utilizing the ribosomes of the host. So viruses do not have ribosomes. They're hacking the system of the host, convincing the host ribosomes to translate their own RNA to produce their own proteins. And then these proteins contribute to new viruses, which are then loaded with the genome. And then what happens next? What's the next step? What? After the package, the viruses have been made anew, what happens next? Yeah, they have to get out. They have to get out. Okay, and there's different strategies for how to get out. Get out. Isn't that like a movie? Okay, they got to get out. What are the strategies for getting out? One strategy is budding. Okay, and budding is what's shown in this picture here, where in this case, if you're envisioning this virus like moving this way, budding is where the virus sort of like assembles itself and then just kind of like peacefully leaves the cell. And as it leaves the cell, in this case, it's picking up some of this outer membrane, and that leads to an enveloped virus. Okay, so budding is kind of like the peaceful way to leave. So many attenuated viruses. What do we mean by that? Attenuated. What would that mean? Attenuated? You guys think vaccines? No, well, I guess attenuated doesn't usually mean killed. It's like weak. Yeah, it's like weakened. In evolution, viruses become attenuated in a sense that we've talked about before. The pathogens in vector biology, they don't want to kill their host because their host is like the car that they're riding in. So if they can kind of attenuate themselves and weaken themselves to the point where they can still replicate, but not the point where they're so infectious that they kill the host, then they sort of have a good deal. So budding is kind of like the peaceful process. Many attenuated viruses, which are weaker, and by weaker, I mean like they're not killing the host, they're not causing significant pathogenesis. They're kind of like just budding out and just continuing their life cycle without disrupting the biology of the host. That's not the only strategy. Of course, there's lysis, which is when if you have a cell and the viruses are replicating inside the cell, the viruses replicate to such a concentration that the cell can literally not hold them anymore, and then the cell bursts and explodes, releasing viruses. Okay, that's lysis. Obviously, that is going to cause pathogenesis, okay, because every single cell that the viruses replicate inside of is going to explode. And if you're exploding the cells of your body, that's a bad deal. Okay, so that's one strategy. And then the third strategy is kind of like the intermediate. Some viruses can go through what's called the lysogenic phase or cycle. And this is where the virus, again, the virus's purpose is to replicate its genome. And if its genome can be converted to DNA, that DNA can get incorporated into the chromosome of the host. And then every time that host cell goes through mitosis, it replicates the DNA of the virus. And the virus is happy, and the mitotic cell is happy because it's not dead. So there's many lysogenic phases in viruses where they just incorporate into the DNA and they just kind of like hang out there. And then different stimuli can cause them to go back into a different infectious cycle. Okay, but they're content to stay there in some cases if the conditions are good. The question is, if the virus incorporates its DNA into the genome of the host, will it go to the offspring? It's a good question. Which cells are the only cells that go to the offspring? Not the stem cells, but sort of. they're called the germ cells. The germ cells. So these are like the testes and the ovaries. And within the testes and the ovaries, there's what's called the stem cell niche, where there's a niche of stem cells, which are the germ cells. And those germ cells are what get passed to the next generation. So if the virus is in a skin cell, are you going to pass it to your children? No. If the virus is in your germline, you're going to pass it to your kids. You can also pass viruses like HIV if they're in the bloodstream, because the bloodstream is shared with the developing embryo. So there are cases where you can transmit viruses from mother to offspring through those mechanisms. But if it's integrated into the DNA, if it's actually integrated physically into the DNA, the only way it's going to get passed to the next generation is if it's integrated specifically into the germ cells. Good question. Please silence that phone. Okay. So just kind of like a brief overview of the diversity here. The genome size of viruses vary to a large extent. So there's large viruses, okay? Large viruses have 2.5 million base pairs. So that's 2.5 MBPs, megabase pairs. That's about, Wolbachia's genome is about 1.3 megabase pairs. So there are some viruses that are double the size of kind of like symbiotic bacteria. So virus genomes can get pretty respectively large. And there's also viruses that are sort of like intermediate, and there are viruses that are small. So like 13,000 base pairs, that's pretty small. You could fit that on a whole plasmid, and many people do that. So viruses can get pretty small or they can get pretty large. There's lots of diversity there. And within vector biology, most of the viruses that we care about in medical entomology are usually pretty small viruses, usually like flaviviruses, which are usually about 10 genes. So most of what we're dealing with are small viruses in medical entomology. I think I just said most of the viruses we're dealing with, like the flavies, have about like 10 ORFs. Okay, so ORFs are open reading frames. What's that mean? We should all know what that means. Yes? Somebody describe it? Yeah, okay, so open reading frame would be, as Ifan said, start codon to stop codon. So most of these viruses have about 10 of these open reading frames. Although it gets a little bit complicated in this organization, we'll talk about in a second. Obviously, this point is that smaller genomes rely much more on their host for replication, for functions, than larger genomes. Larger genomes are going to give you a bigger repertoire of things that you can do, bigger toolkit. But it's also going to be slower for you to replicate. It's going to cost more energy for you to replicate. So there's kind of like a trade-off between versatility and streamlining. Okay. Here's where it starts to get confusing. Many different flavors. But it's simple in a sense if you just understand the basic gist. The basic gist of virus genomes is that they're either DNA or RNA. Okay, some are DNA, some are RNA. And of course, nucleic acids can either be single-stranded or double-stranded. Okay, so many virus genomes, pick one and then pick another. You can find different combinations. Okay, so some viruses have DNA, some viruses have RNA, some viruses have single-stranded RNA, some viruses have double-stranded RNA. And then within that, there's another variable, right, which we should know, is that DNA has an actual, it's a vector in a sense that it has direction. DNA has directionality. And usually what we think of as 5' to 3', this is the sense strand of DNA. Sense strand, what's unique about it?
It's not actually the strand that actually makes it. It's the lagging strand, right?
That's not what I'm thinking. What's the important thing about 5' to 3' sense positive RNA? What's the important thing?
Is that the way the polymerase reads?
That's also important, and I will talk about that. But I'm thinking even more crucial, more critical, more basal.
If it's in the session, then it doesn't have to like meet the,
What is messenger RNA?
it's like the template to make protein.
It's the immediate template to make proteins. What is the directionality of messenger RNA? Incorrect.
No, wait. The
I would really hope that you would know that by now.
The antisense that makes the stuff. It's the antisense that is actually responsible for.
The antisense would be the code for the messenger RNA.
Correct. So that
But the
means-
messenger RNA is what direction?
5 prime.
Thank you. Messenger RNA is 5' to 3'. That means from 5' to 3' RNA molecules can literally be immediately loaded into the ribosome and immediately translated into protein. So viruses that have sense single-stranded RNA are special in a sense that they can immediately inject their genome and it will immediately be translated. Other viruses have issues in that they have to go through conversion process and convert their DNA or their RNA to something that can be made in proteins. So there's often a conversion process in viral genomes if they're not 5' to 3' sense RNA. Did you have a question?
No, I was going to say, like, this is what Dr. Martin said.
Yes.
TWSB.
Yes, this is exactly what Dr. Martin emphasizes because she studies viruses. Just for a brief overview, because clearly we were not as up to date as I would hope. 5' to 3' directionality is designated by the carbons in the phosphate backbone that make up the nucleic acids that, or the nucleotides that go into the nucleic acids, okay? So if you look at the carbons on the backbone, they're labeled 1, 2, 3, 4, 5. 5 means it's going from here down to here, and the linkage of the nucleotides is going from 5 to 3, okay? So that's where the 5 to 3 comes from. And again, just for a brief review here, this is like a little trick I use. I know some people have seen this before. Again, I just repeat it because it's even often difficult for me sometimes. So when I'm trying to envision the directionality of DNA, I usually draw this triangle, and the side I write as the 5 because it's higher up than the 3. And I just try to remember that ribosomes read downhill. So ribosomes read downhill. They're big and they're kind of lumpy. And if you just put them on this, they would like roll down the hill, right? That means the 5 to 3' molecule can literally get loaded into the ribosome and translated immediately into protein. Polymerases are less lazy than the ribosome. Okay, bad scientific lingo, but just a trick to help you try to remember. Polymerases have to go up. They read 3 to 5, but they write 5 to 3. Okay, so that's kind of a trick. This is important. It's very important, especially when you start to get to molecular biology, right, Lewis? Directionality of the DNA is extremely important. When you actually have to start like designing primers and stuff, it essentially becomes like an everyday thing that you deal with. So you really want to spend time kind of like looking at this. Now, viruses often have a very special enzyme, and I talk about this enzyme in my biotechnology class because it's a very special and important enzyme. And it's also special and important for the virus life cycle. Because as I just said, if the virus is 5 to 3, it can just inject that 5 to 3 genome into the host cell, and then that can be immediately made into proteins. But if the virus genome is anything other than that, it often has to go through a conversion process, the genome. So there's this enzyme called reverse transcriptase. And reverse transcriptase reverses the central dogma of biology because reverse transcriptase can take messenger RNA and it can convert it to DNA. Okay, essentially. It takes RNA and it can write DNA matching DNA, which we call complementary cDNA. So if I were to draw this out, if this was the 5 to 3' RNA molecule, the reverse transcriptase can write, I'll get a different color, write a matching DNA sequence, which can match. And now we have, this is 3. And now we call this thing cDNA, complementary DNA. It's complementary to the RNA. And this complementary DNA can often go through another conversion process, and that is what often gets integrated into the genomes if it goes into the lysogenic phase of the virus. Okay, but you can't integrate RNA into the genome, right? RNA is not just going to randomly like insert into DNA. They're two completely different molecules. Okay, so oftentimes reverse transcriptase is necessary for that process, and oftentimes if the genome of the virus is not 5 to 3, if it was 3 to 5 single-stranded RNA, oftentimes this needs to be converted to 5 to 3 RNA, and that can also happen with an RNA-dependent RNA polymerase, and I think it would be also called a reverse transcriptase. Essentially what you want to know is, again, like the structure of the virus genome is very important, and there's often a conversion process that has to happen for the virus to complete its life cycle. And that is sort of the central focus of much of virology. Okay, I went through this. Okay, so here I have antisense RNA genomes. That means the virus genome looks like this. So if this is a virus, crystal type of virus, if it's antisense genome, oftentimes inside of the virion, reverse transcriptase will get packaged into the virus. So sometimes viruses are loading you up with not just the genome, but also enzymes that are preloaded, ready to fire, so that when that virus genome is inserted, the reverse transcriptase can immediately go through the conversion process. Whereas if you didn't load that in, you'd have to rely on the host cell to somehow convert this into some functional messenger RNA. That's never going to happen. So in cases where the genome is not immediately translatable by the ribosome, the virus often preloads a prepackaged reverse transcriptase as like a firing pin to execute the conversion immediately when it happens. Flavies, well, I won't go into it now, lest I make an error. Yeah, I already said this. Sense RNA viral genomes can immediately be translated. We get the picture. Okay. I included this blown glass structure. Have you guys, there's an artist who does things like this, but they look like viruses. So this is a pretty lecture. I already said this. Some viruses, if they're single-stranded DNA, have to make double-stranded DNA. That's another conversion that sometimes has to happen. Again, just another emphasis of this conversion process. Okay, so the way that viruses are taxonomically classified is by the structure of their genome. So certain clades of viruses are recognized as DNA viruses. Certain clades are recognized as RNA viruses. RNA viruses are split up into single-stranded versus double-stranded, et cetera, et cetera, et cetera. The ones we care about, which I'm going to talk about further, are the Bunyaviruses, the Flaviviruses, and the Togaviruses, because these are ones that are transmitted by vectors. And I'll talk a little bit about this because you could ask yourself a question of, okay, so you got a host, you've replicated a bunch of virus genomes, you've replicated a bunch of virus proteins. Many people study the question of how does a virus actually assemble? Like what puts the virus together? What builds the machine? And how does that process happen? And of course, depending on the virus, that process is filled with much diversity. Okay, so in one case, the assembly is spontaneous. And that means you sort of build up, you program pieces of a larger whole that are kind of unstable by themselves. But if they find each other, they bind and sort of produce a more stable state. So if this is the case, the virus can sort of fall down the scale of entropy because things become more stable in this case, and sort of just fall down. It doesn't cost any energy to assemble. They just kind of find each other and spontaneously assemble. Okay. And if that happens, so imagine you have a capsid. Here's a capsid that's spontaneously assembled. You often ask the question, and it depends on the virus, now does the genome get squirted into the capsid, or did the spontaneous assembly happen randomly around the genome and the genome is already captured inside? These are two different variants. Sometimes it's assembled around the genome, and sometimes the genome is like loaded in. Okay, these are two different things. And oftentimes it's not spontaneous. Oftentimes there are proteins that use energy to assemble the virus. So this would be the opposite of sort of like a spontaneous assembly. More sort of like common like virus nomenclature. If you see a protein that is called S, many viruses have these like strange, oh, I guess not strange, but they're like alphabetical names of proteins. These usually mean something. So S usually means structural proteins. That's usually like things that form the capsid. So here's a picture of a spontaneous assembly around a genome. I guess this would be something similar. NS, of course, non-structural protein. So you might see NS in reference to a reverse transcriptase because it's a non-structural protein. Talked about that. We already talked about these things. Flaviviruses, again, which are going to become very important for us. Flaviviruses are enveloped, and they get their envelope from budding. And I think I show this picture to show you that the envelope around the virus doesn't always come from the same place. Doesn't always come from the outer membrane, okay? In some cases, the virus might be being assembled or go through a process where it goes through the Golgi, and then it might pick up an envelope from the Golgi. And then as it comes out, it might pick up a second layer of envelope from the bilayer, okay? So many viruses pick up different envelopes from different parts of the cell. So just keep that in mind. It's going to depend on the virus. More terminology. Arboviruses. That's very important for us to know what this means. This is a catch-all, a short term for arthropod-borne viruses. These are viruses that are transmitted by insect or arthropod vectors. So we care mostly about the arboviruses. Most of the arboviruses are single-stranded RNA. It's actually pretty easy to remember. The ones we care about are single-stranded RNA for the most part. RNA viruses mutate rapidly because they don't have self-correcting mechanisms against a template. If you're single-stranded, right, let's compare this, I guess, to double-stranded. In a situation where you have DNA and there's a mutation, there's a second site from which proofreading enzymes can fix that site. Does that make sense? If you're a virus and you're single-stranded and you get a mutation, there's no template to repair that from. So this is one functional mechanism of why viruses mutate so rapidly. There's no proofreading mechanism in single-stranded viruses. Why would the virus want to mutate? Yeah, adaptation. It's gonna foment spontaneous adaptation. And viruses are famous for being quick adapters. Okay, introduce these. There's alpha viruses, flaviviruses, and bunyaviruses. We'll talk more about these, but quick, alpha viruses. Chikungunya is a famous alpha virus. Yellow fever, dengue, Zika are flaviviruses and buniviruses. I think I give some examples in the next lecture. Most of these viruses, these short RNA viruses, encode about 10 proteins. Okay, this is what's unique about arboviruses, and this is a very important point. Remember at the beginning of the lecture where I told you bacteriophages, they only infect bacteria, and viruses are very specialized. This is true. Arboviruses are very unique. Arboviruses can replicate in more than one host. Which hosts might arboviruses replicate in? Arthropods. What else? Arboviruses, shut that damn phone off. Arboviruses are famous for the zoonotic cycles. And in the zoonotic disease cycle, there's what? This was just on the test. There's the reservoir. Exactly. There's a reservoir. So arboviruses are very unique in that they can replicate in arthropods and also in the reservoir. And these are two very different types of cells. Arthropods are going to be like the insects, like the mosquito cells. And the reservoir is going to be like vertebrates, like maybe like cows, maybe like humans, okay? Maybe like monkeys. So to be able to be a virus that can replicate in both mosquito cells and like specific mammalian cells is very unique. There's not very many arboviruses, but the ones that can do this are the ones that are very dangerous. So arboviruses can replicate in both the vertebrate and the arthropod hosts. This is very unique. It's a very significant accomplishment in evolution. In terms of arbovirus protein nomenclature, you'll see glycoproteins notated as GN and GC. So it means glycoprotein, the G. Protein N would be non-structural, I assume, and in this case, the transcriptase is labeled as an L. That's not super important, but just know that this, I kind of show you this just to show you that like viruses have these weird protein names that are just sort of like alphabetic letters, which seems like completely useless in some sense, unless you know the code. Okay. Okay, now I go through sort of some other viruses you might hear of, especially with respect to entomology. So baculoviruses are a family of viruses, and they're very important for biotechnology. These are, now I gotta yell at myself, shut that fucking phone. Baculoviruses are very famous for biotechnology, and these are sort of been developed as vectors to express recombinant proteins in insect cell cultures. Okay, so baculoviruses are a very, very famous family of insect viruses, and they've been hijacked, or I guess hacked, to be used in biotechnology. And one of the reasons these are so popular is because many of the popular recombinant protein expression cell lines are like SF9 cells, which are butterfly cells, or S2 cells, which are Drosophila cells. The baculoviruses are essentially like, kind of like a system of plasmids, although they're not plasmids, but a system that through which you can express recombinant proteins in insect cell culture. So I show these here just so that like you've heard of this. If somebody says like baculovirus, in the insect world, you want to know like what baculovirus is. These are circular double-stranded DNA genomes. These are not arboviruses. Remember, arboviruses are special arthropod-borne viruses that can replicate in both a reservoir and a host, or and a vector. Baculoviruses are not like that. Buniviruses. Buniviruses are very unique, and they're kind of like the most, I guess, the most weird. They're not flaviviruses, but they're the most weird sort of like arboviruses. They have 3 different pieces of genome. So they have 3 different, they kind of look like little plasmids. They're single-stranded, negative sense RNA molecules, and they're not circularized, but a hydrogen bond forms them into a closed loop. That would mean that if this was the molecule, there would be like an A here and a T here, and then they would find each other, and that would form what we would call a hydrogen bond closed loop. Does that make sense, Ifan? So they're not closed like a plasmid. They hydrogen bond and form these closed loops. and then these loops can be twisted. They're enveloped. And this is a very, very common theme in arboviruses, which becomes very, very important. One of these segments encodes what we call a polyprotein. A polyprotein is kind of like an operon, but like the virus version. A polyprotein means there's a start codon, there's a stop codon, and let's say this is 10,000 base pairs. This is like a giant ORF, a giant ORF. So a polyprotein involves a giant ORF, giant open reading frame. And what happens is when the ribosome hits this, it feeds it through and it translates a giant protein, which we call the polyprotein. And in this protein are little sections. Let's call this like section 1, section 2, section 3, section 4, section 5. And often in this polyprotein, at the end or in the beginning or somewhere in the middle is a special protein called a protease. What do proteases do? They cut proteins. So if one part of this big protein is a protease, oftentimes it will begin to cut itself up, cuts itself into little pieces. Of course, where does it cut? It cuts at the specified sections. And so then what gets produced is, in this case, seven different, 1, 2, 3, 4, 5, 6, 7 different proteins, but they were all derived from the mother polyprotein. Does that make sense? So the key here is there aren't different genes, there aren't different promoters, there aren't different regulatory circuits. There's one promoter, one ORF, which gets translated into a giant polyprotein, and the polyprotein cleaves itself up into smaller segments, which constitute the smaller proteins. This is a very common pattern in arboviruses. So bunyaviruses do this, and flavies also do this. So the important things to remember about the bunyaviruses is that there's 3 pieces. They form these loops. It's single-stranded negative RNA, and one of these is a giant polyprotein. Here's the process by which I just described. Here's the polyprotein, and there's a protease that usually cuts this thing up into little pieces, and then these pieces constitute the separate proteins of the virus. 2 minutes left. Oh my, I'll skip that. Half of all arboviruses are bunyaviruses. There's a lot of these. Mosquitoes vector 42 of these, ticks vector 2. So the vast majority of the vectors for viruses are mosquitoes. That's why one reason why we care so much about mosquitoes. Dr. Martin talked about this. Different segments of the genome can often exchange with other segments of other viruses, and you can get these like chimerics, and that can cause problems if you're co-infected by 2 viruses at the same time. It can cause problems. Examples of bunyaviruses are La Crosse encephalitis, Crimean Congo hemorrhagic fever, Rift Valley fever. I talk more about those later. Flaviviruses. Flaviviruses are extremely important. This is the next family. These are positive single-stranded RNA viruses, about 10 to 11 kb, and they also form the polyprotein. So they're different from the bunyavirus in the sense that they're positive. That's how they're different. But they also do the polyprotein like the bunyaviruses. The flaviviruses, the famous flavies are what? Zika, dengue, yellow fever. These are the famous flavies. Flavies are famous for they get in your cells and they melt your internal organs and you essentially dissolve your insides and then you die. Those are what we call the hemorrhagic fevers. Dengue is famous for that in dengue shock syndrome. Zika, not so much. Yellow fever is famous for melting your insides. Flaviviruses are unique in that it's interesting, the phylogeny of the flavies actually matches the vector that spreads them. So it's highly unlikely that a mosquito just picks up like a random flavy and spreads it. The spreading of these arthropod-borne viruses is very evolutionary adapted. So Culex, for instance, vectors a very specific subset of flavies. Aedes, which vector the hemorrhagic fevers, vectors a very specific subset of these viruses. There's a tick-borne clade, which is vectored by ticks, and then there's other things. Okay, but the phylogeny correlates with the species that spreads it. This means that it's a highly evolved thing. It doesn't just happen. It doesn't just all of a sudden like in viruses we worry about something like what we call jumping. It just jumps to a new host. It's very, very rare. It's very hard to do that. And this is the proof of that the phylogeny correlates with the vectors. Togaviruses, this is the last one that we talk about in this lecture. Single-stranded positive genome like the flavies. They're enveloped, and the famous examples of this are chikungunya, young virus, and Western equine encephalitis. Symbis is a very famous model for togaviruses, and we use that in Drosophila if you wanted to like model systems of togaviruses. Here's a virus on this lamb's tongue. There's other things. Mostly what we think about are the togaviruses, the flaviviruses, and the bunyaviruses. I'll finish quick here, but I do want to hit these points. Oftentimes, there's a very special reservoir called the amplifying host. Sometimes just having the virus as a reservoir doesn't mean you're like the main reservoir. Oftentimes, there's a reservoir which is really where the virus is like the viremia of the virus in that reservoir is going to like extreme levels. That's called the amplifying host, okay? And it's amplifying like the infected proportion of the arthropods. So that's a particular reservoir where the arthropods are just honing in on that reservoir, picking up virus at high levels, and then that's what's amplifying the virus in the vector population. That's the amplifying host. Competent host just means it's a host that if the virus gets in there, it can replicate. And it can, in theory, like transmit it to an arthropod. What was the question? Okay, we already know this. So there's a concept called reservoir competence, similar to the vector competence, in that some reservoirs are particularly good at like setting this, increasing the viremia and getting this loaded into the arthropod. There's actually a specific equation for each of these particular diseases and each of the vectors that spread these diseases, which determines the vector capacity. So for each mosquito that spreads one of these viruses, you can calculate an actual like quantitative value for corresponding to the vectorial capacity. And things that go into this equation are how many mosquitoes there are per person, the bite rate, like how many average bites an average person gets, survival rate, like how long they survive. It's the older mosquitoes that are more dangerous, the extrinsic incubation period, which we talked about, and natural log, we know about that. So essentially I'm telling you there's just actually like a formula that you can calculate like how good something is at spreading a virus. And a vector, I should have given this in the first lecture, but I give it here. A vector means a very specific subset of things. You can't just say something is a vector. And this is the subset of things that being a vector means, okay? It means that somebody has done the experiment and isolated the virus from that species. So for instance, they've gotten a mosquito, they've extracted the virus at the minimum, a certain minimum threshold rate of detection in that mosquito, okay? So it's detection, it's there. Not only that, people tend to think that's the only thing you need to be a vector. No, there's other things. The host preference of that mosquito has to include the amplifying host. So you have to identify what's the amplifying host, and you have to validate that that mosquito actually goes to that host and picks up the virus from there. There has to be spatial overlap with the host, like geographically, they have to be in the same region. The feeding behavior has to overlap. So like we talked about how filarial worms would get picked up at night with the culex because the feeding behavior overlapped. And then there has to be a laboratory demonstration that the vector can actually transmit from A to B. So these are the things that have been proven when we say this is a vector. Last slide here. Different mechanisms of transmission for viruses. This is just important to keep in mind. Venereal transmission, that's going to be like sexually transmitted viruses. This can actually come into play in the case of Zika, where people were worried about if a male mosquito can actually like transmit. Well, in one case, it's venereally transmitted in the humans, I should say. Like I think it's been demonstrated that humans can, through sex, can transmit Zika virus. Is that correct? I should be careful here. I think there's some studies. That was a worry. That's a good way to say it. That was a worry. But there's also a worry from the arthropod side, from the sense of if a mom has it or if a dad has a virus that they somehow picked up, can they sexually transmit it when they mate? That's also like a worry in the arthropod side. Vertical transmission would be mom to children. Horizontal transmission is when the vector acts as a vector and it's transmitting from reservoir to a new thing. Mechanical transmission would be like a dirty needle. That's different from what mosquitoes do. Aerosol transmission, which is what we were about with coronavirus, it's in the air and it's like you're getting like the particles. Flaviviruses are not aerosol transmitted. So these are important things to know. And then another means of transmission is contact with like infected tissues or blood. This is famous for like Ebola. Ebola gets in like the human body fluids, and that's usually how it's transmitted. Okay, so that's sort of the end. This is like a basic overview of virology. And then the next lecture I have is sort of like, here's the famous arthropod-borne diseases. Okay, thanks for staying 7 minutes late. It can okay, good. I was fact checked. Thank you. Did you have a question?